So thank you for the introduction. This is my first time presenting in Puerto Rico and my first time presenting with an Apple computer. So we'll see how that goes. So I actually have several different roles in San Antonio. I'm the director of the Proteomics core, the Biomarkers core, and I do my own research mostly on pediatric disease, so brain injury, acute lymphoblastic leukemia, and lots of collaborations with other researchers, study everything from multiple sclerosis to ant behavior, believe it or not. So I thought I'd begin today just a little bit about my background, talk to you about tandem mass spectrometry and some of the analytical challenges that we face as chemists trying to use this technology to find biomarkers. Most of the publications that are out there for microwave and magnetic proteomics have to do with protein biomarkers of multiple sclerosis, and I'll talk about the technique last. So please interrupt me if you have any questions. I think I've only planned for about 30-35 minutes. So as Lloyd had mentioned, I've worked in lots of different places, right? And both in industry, national lab, and universities. If there is a theme here, it's really that I've used multidimensional techniques to study proteins. And so any analytical technique, you improve the selectivity with the more dimensions of analysis. So throughout my work, you'll see I did LC, LCMS, LCMSMS, LCMSMSMS. It just keeps going. So unfortunately, the more hyphens you have, the more complicated the technique, and the more difficult it is to keep it working all the time. So I've become pretty good at troubleshooting. There's one thing I'm not talking about at the very bottom, actually two. So recently, just to try something new, we've gotten into doing some nanomedicine research. So we're actually building our own nanoparticles that have cancer drugs, as well as some potential to make some drugs for multiple sclerosis or brain injury. And that's actually something I'll talk about some other time. And then we've also looked into writing new computer algorithms. So unlike microarray and a lot of the genetic techniques, proteomics is more complicated. There's 20 amino acids, hundreds of post-translational modifications. And then the technology isn't really a platform technology yet. You can't just buy like an Affymetrix chip and collect the data in one way, and you can't analyze the data in one way. So this year I took the plunge and started writing my own algorithms with an electrical engineering professor. And it's been challenging. I got humbled by how little math I know, but it's been a useful adventure. And so a lot of the things that I'm going to show today we've since automated with some computer algorithms written in the stats package called R. So I actually like Wikipedia. Do you guys like Wikipedia? No, okay. So I've heard it's not scientific, but here it is. What's a biomarker? There's several different kinds of biomarkers, and this word is abused in literature for lots of different reasons. We focus on diagnostic, prognostic, or predictive biomarkers usually, and I focus on protein biomarkers. So right now there's actually less than 100 proteins measured in your blood if you go to the doctor, despite the fact that we know there's 30,000 proteins in your cells. And so proteomics can measure between 5 and 10,000 proteins if you really push the limit. So why aren't we measuring more proteins in our blood? That's because there haven't been enough studies that can correlate the level of any particular protein with a disease, or there haven't been enough careful studies. And just like I said that proteomics is behind microarray in the technology, the ability to run lots of clinical samples to get enough data to get statistically significant correlations is challenging. So what you find is most proteomics experiments generate long lists of proteins in a very short number of samples. And so what I tried to do in the last 5 years is do the opposite. So we try to generate maybe a shorter list of proteins in a large number of samples so we can build up the statistical proof that that is actually a biomarker for disease. And we're focused on diagnostic or prognostic markers. So an early detection biomarker is one where you can measure a change in the protein level before there's some clinical observable. So I'll show you data later where we can measure proteins, for example, at day 7 in this mouse model of disease, but you don't see any measure of paralysis or clinical measure until day 10. So this would be an early biomarker of detection. And in the pharmaceutical industry, of course, the emphasis is on predictive markers, markers that will let them know, are you going to be a good responder or a poor responder to a particular drug? And this is where probably the field is going to go for a long time. So we're funded for an RCMI grant to actually look for predictive markers of glucocorticoid resistance in an experimental autoimmune encephalomyelitis mouse model of multiple sclerosis. So we're actually just now, 3 years in, treating the mice with glucocorticoids and seeing how their protein expression profiles change compared to, the untreated controls. But we think we found some diagnostic and prognostic markers. How do I dim the lights a little bit? Oh, thanks a lot. So this is actually a slide I made when I was a grad student, and I'm proud to say I'm still using it. So this is,
Thank you.
does anybody know what this is? So the first neuropeptides measured by Desiderio, I think in the 80s, by mass spectrometry are leucine enkephalin and methionine enkephalin, very important neuropeptides. And if you look at the structures of the two, the sequence is almost identical. It has one different amino acid at the C-terminus. And so how do you distinguish the sequence of these things with a mass spectrometer? So if you just measure the mass of methionine enkephalin is 574 Daltons. The mass of leucine enkephalin is 556 Daltons. So you could distinguish them just based on their mass. But if you had a mixture of 10,000 peptides, if you were to measure all the peptides in serum, for example, you can imagine scrambling the sequence and you get the same mass, and you wouldn't be able to unambiguously quantify one peptide just by measuring its mass. So everything I do is what's called tandem mass spectrometry, or MSMS. So we measure the mass, sorry, does anybody have a laser pointer or not? Thanks. We measure the mass of the peptide as well as the mass of all of the pieces of that peptide if we break it in the gas phase. Oh, fair enough. That's okay. So you can imagine, in proteomics, the common thing to do is you take the protein and you digest it into peptides with an enzyme. And so then your protein might give you 10 peptide fragments. We have it. Okay. I don't know if that's going to work. It's okay. Let's just keep going. So if you take a protein, you digest it into peptide fragments, you can try to understand what happened at the protein level by measuring those peptides. Thank you. In the same way, you can take a peptide like methionine or leucine enkephalin and digest it or cleave it in the gas phase into pieces. So if we cleave this peptide along its backbone, you'll generate pieces with different masses. And so if the pieces of the peptide that are measured keep the N-terminal amino acid, we call them A, B, and C ions. And if they keep the C-terminus, we call them X, Y, and Z ions. And you establish the sequence by building up a ladder of masses from either direction. And so what's important is not many technologies that measure proteins are actually sequence specific, but tandem mass spectrometry is. So just like there's a trend in genomics to sequence everything with next-gen sequencing technologies, it's the same in proteomics. So actually the field of analytical chemistry, I think, recognized this pretty early on because many of us saw this technique and stuck with it. There's a lot of other choices at that time. So I got actually one of the first PhDs in this field called proteomics, which is built on tandem mass spectrometry. One of the other things that happened around the time I was getting my PhD is there was 2 Nobel Prizes given. One for interfacing the ionization method, or the way to convert the peptide from the liquid phase to the gas phase, is called electrospray ionization. And it turned out that to do that, it was useful to work at really low flow rates. And so what happened is the lab I got my PhD in with Dr. Kennedy, they're experts in what's called capillary liquid chromatography. So if you've ever done HPLC, it's usually with a large bore column, like a 4.6 millimeter column that runs at 1 milliliter a minute. And that's typically what's done in a pharmaceutical company. In academic circles, people did miniaturized capillary liquid chromatography for analytical work. And it wasn't really very useful for mass spectrometry. But it turns out that if you do very low flow rate capillary liquid chromatography, it's easy to ionize all of the effluent from the capillary LC column and send it into a mass spectrometer. So all of a sudden, the technique that my PhD advisor was an expert in became super important for mass spectrometry. And he recruited me from Indiana because he'd been a chromatographer his entire life and had lots of peaks, and he wanted to know what they were. And so there's this line that if you're a chromatographer, the worst thing is having unknown peaks, and if you're a mass spectrometer, it's just having peaks with masses. So we like to sequence everything now. So we get the peak, we get the mass, and then we get the masses of the pieces of that peptide and sequence it. So he was a neuroscientist and said, Will, I've got all this data that I've collected over 10 years studying rat models of different neuro diseases. Tell me what all those molecules are, basically. So no big challenge. And so there's some things to keep in mind for the rest of the talk. The capillary LC columns have an inner diameter smaller than a hair on your head. So they have very low capacity. That means we can only inject 10 to the minus 12 picomoles, a very small amount of material onto the column. So everything's miniaturized compared to what's normally done in the laboratory. And then tandem mass spectrometry only has a dynamic range of 3 to 6 orders of magnitude. So what does this mean? If you're studying blood, where the proteins vary in concentration 12 orders of magnitude, and you can only load a picomole of material, you're stuck with just measuring a few thousand molecules or maybe 1 million molecules in your mass spectrometer. So that means that the sensitivity of the technique needed to be dramatically improved to make it something that could be a research tool, like a fluorescence or something like that. So capillary liquid chromatography with fluorescence detection was the state of the art just 15 years ago, I think. And that's the way you did a lot of research. So the challenge for my PhD was really identify things at femtomole to attomole levels in the presence of higher abundance peptides or proteins. So one of the other challenges of the technique is if you have some high abundance molecules, they mask or block the measurement of the low abundance ones. And you see this in genomics too. So that takes me to this project I'm going to use as an example today. In any analytical technique, there's this famous triangle that the method is a compromise between sensitivity, selectivity, and analysis time. And you can't have all three. So one of the things that I wanted to do, as I said, was measure a large number of samples, maybe not so many proteins per sample. So I developed this method called microwave and magnetic proteomics that reduced the analysis time without sacrificing the sensitivity or the selectivity. And for this particular project, there's no biomarkers of multiple sclerosis. So we're starting at ground zero. And so we wanted to measure several thousand proteins simultaneously in brain tissue or CSF or blood in a longitudinal sample. So it's a time course. And then we wanted to not just measure them, but discover or sequence those markers, right? So we're basically identifying and quantifying everything at the same time. And multiple sclerosis, like brain injury, is actually a disease of demyelination. So you have T cells infiltrating the brain, and if you take a look at a neuron, the myelin sheath can be demyelinated, and debris accumulates around the sheath, forming these lesions in your brain. And so there's controversy on whether or not macrophages or microglia are beneficial or harmful to that demyelination event. So we expected that some of the good biomarkers would be myelin-specific proteins or proteins involved in phagocytosis, either by microglia or macrophages. And so on top of those things, I run a core lab, so I tried to approach this problem and the other problems that we were dealing with very systematically and try to find what the bottleneck would be to accomplish the goals of this project and every other project. And so we kind of broke all the projects up into 6 steps. We have kind of an experiment design step where we think about what's the best number of samples needed for the particular power of study. We might do some labeling. I'm not going to explain much about this, but we do a lot of what's called isobaric labeling, which I'll explain later in the lab. And there could be some low-level proteins that are essential for the investigator to measure to publish. So for multiple sclerosis, we thought we better measure myelin basic protein, for example. And if we weren't able to measure it, then we might need to do some immunoenrichment steps. Then we do this technique, which I mentioned a little bit. The technique basically separates peptides in time by capillary liquid chromatography, and then measures the mass of the peptide and its fragments by tandem mass spectrometry. So having the masses of the pieces, just like I showed you for methionine and leucine and enkephalin, doesn't really help you if you collected 20,000 MSMS spectra in an hour. So when I was starting out, it would take me about an hour to get a sequence by hand, by looking at the mass spectrum. So there's no way you can keep up. So the way this is done nowadays is you search all the data, which might be 20,000 peptides worth of MSMS spectra, against a database of all theoretical MSMS spectra from all known proteins in the organism you're studying. So in our case, it's mice. And so you can imagine, for example, if we did this technique and we measured a mass at 556 and another at 574, we might think we have methionine or leucine and enkephalin in the sample. And then by searching the database against all those fragment ions, we could assign which one it was. The next step is statistical analysis. So if you write a grant on biomarkers, the first thing the reviewers are going to ask you is, how do you prioritize the markers that you found? How do you know these are the most important proteins in the disease? And so through statistical analysis, we can correlate the protein levels or the peptide levels to some other measure. So a lot of times we will correlate simply with times. You can correlate to the disease course, or in this case also the EAE score, which is a measure of the paralysis in the animal. And so everything I'm doing is a correlative study. The p-values aren't simply the fold changes from the protein. They're the fold changes combined with the time measures and some clinical measure, because we always want to correlate back. And then finally we map to some kind of biochemical pathways. We use this program Ingenuity to try to link our data with all the other data that's out there. So it's a big knowledge base of pathways in literature, and sometimes this helps us pick more upstream targets or downstream targets. So now I'm going to talk to you about the M2 proteomics itself. I think, let me say if that's right. Yeah. So this is kind of the workflow for sample preparation and explains the title of my talk. So if you had to prepare 160 samples quickly, it would be a nightmare to run 2D gels. So usually do 5 to 10 replicates per mouse. So you're looking at hundreds and hundreds of gels, and they're not very reproducible unless you really know what you're doing. So we came up with a gel-free method called microwave and magnetic proteomics, which basically takes the proteins, incubates them with magnetic beads with C8 chains on them, and the proteins simply stick through their hydrophobic groups onto these magnetic beads. We then do on-bead reduction, alkylation, and digestion to unfold the proteins, break their disulfide bonds. And because the proteins are bound to the bead while we're doing it, we have very high sensitivity. Normally this is done like in a gel slice or in a tube, and a lot of the protein sticks to the side of the tube or into the gel slice. Then we add trypsin and cleave it into peptides. So the proteins that are bound to the bead, if you add a protease, the peptides will fall off the bead and will collect the supernatant. So now we've generated peptides in the lab from proteins that we had reduced and alkylated on bead. And if you can imagine, we treated 5 different samples this way. We could label each one of those samples with a different color reagent. So in this case, we're labeling it with a blue reagent. And you can imagine some other samples labeled with a yellow, green, red, or white reagent. The advantage of this is then you can combine the samples into one sample mixture and analyze them simultaneously. And this is how we do protein quantitation quite often in proteomics. It's relative to everything else. So the protein expression of the mice that were labeled with the white reagent, for example, here at day 10, will be compared, let's say the red reagent, will be compared to the mice labeled with the blue reagent, or the yellow, or the green reagent. So they're not really colors. This is mass spectrometry, and I'll talk to you later what the labeling reagents actually are. So to make this quick, we found that we could use a microwave. So microwaves have been used in organic chemistry for, 10, 20 years pretty routinely. In proteomics, not so much. So how many of you microwave your food at home? How many of you use a microwave in the lab? So it's actually more than I thought. So at UTSA, nobody raises their hand except me for the second question. And it's silly. So anything that's an aqueous buffer, you can accelerate the reaction in the microwave, including enzymes. So what does this mean? For us, this meant a 30-fold improvement in our sample prep time. It took us 30 times less time to prep a single sample than it did using gels. So that made these 160 mouse experiments or 500 leukemia patient experiments feasible. So for the first time, we could actually have the time to prepare enough samples to collect the proteomics data. And I get a lot of questions at conferences like, is this as efficient as other methods like overnight digestions? And how do you know this is? So we have tons of statistical data, so it works is the bottom line. I didn't do a long study of it. One of my colleagues at Genentech, Jenny Lill, also uses microwaves routinely. So this is something that's going to come into common practice, I think. So how do you compare? Let's say we have 5 samples, we mix them together, we make one mixture. So then if we do LC-MS, we're going to get protein expression for every sample. How do we combine more than 5 samples? So this is actually the disease time course for a mouse model multiple sclerosis. You can see at day 20, the disease scores peaks around 2.5. So these are the mice with the worst disease, or the peak of demyelination. And so you can imagine if we took 4 mice from day 20 and we pooled the material and labeled that pool with the white reagent, we could then mix that back into all the other time points. And then as long as at any other time point, let's say these 4 mice, we express their protein levels relative to the pooled reference, then we can compare across all time points. So what we did basically is we made pooled reference material and blended it back into each of the time points we could normalize by time. So this is a way that you can take a 5-plex experiment, and turn it into a 50-plex experiment. You just need to use one of those encoding reagents as pooled reference. And there's a lot of tricks on what's the best way to do that. So earlier I showed you the MS-MS spectrum for methionine and leucine encapsulin and how we get the sequence. So here's another example. This is a peptide, I think it's from albumin. It has this amino acid sequence. And now we've labeled, for example, the B1 ion means the peptide was cleaved there, B2 is there. And you can see that we can establish the sequence as we cleave along the peptide backbone. So how do we quantify? What's really spectacular about these tandem mass tag reagents is what they're called, or isobaric labeling reagents, is instead of 5 colors, what we have is 5 different reagents with this structure. And they're called isobaric because they all have the same mass. So it's kind of counterintuitive. Why would you take 5 different samples and label them with a reagent that all has the same mass and then do mass spectrometry? It doesn't make any sense. Like, how are you going to distinguish them? So it turns out these reagents were designed so that when they modify the amino termini of lysine residues or the amino terminus of the peptide, they all get a shift of, I think it's 229 Daltons. So you can imagine this peptide has, let's just say it has a mass of 1,000. If you derivatize it with a reagent like this, you're going to add 229 Daltons to the lysine residue for the reagent and 229 to the amino terminus. If you had 5 other samples and you use different reagents, every one of those would have that sequence and those masses. But when you do MS-MS, that is when you cleave this along the backbone, you get what's called reporter ions. So in the very low mass region, which is expanded here, you get the different reagents. So one of the reagents gets cleaved at, actually they all get cleaved at this position, but we've put C13 or nitrogen 15 atoms in the molecule. So you can imagine it's one reagent, and then if you use carbon 13 or nitrogen 15 to shift the position of the heavy atoms, when you cleave the reagent, you'll get these different reporter ions. And so one of the reagents cleaves to give you a reporter ion at 126, another's 127, 128, 129, 130, and 131. So these are called the 6-plex tandem mass tag reagents. They all have this structure with different C13 and nitrogen 15 atoms placed in. And what's beautiful about the technique, unlike a lot of other proteomics methods, you can imagine that I told you the capacity of the capillary LC column is very small. You can't load much protein on it. So if you had 6 samples, it's like you're loading 1/6 the material from any particular sample if you had mixed them together. If you do these techniques, because all of the peptides from the different animals have the same mass, all these sequence ions are actually the sum of 5 different animals. So that B2 ion is actually the sum of the B2 ions from 5 different animals. So what does this mean? Practically, it means that we can multiplex or put as many animals, proteins together as we want, and it won't hurt our ability to establish the sequence of the peptide. And then to do quantitation, we just take the ratio of those reporter ions, just the intensity. So here, the mouse that got derivatized with the 130 reagent, you could say is 2 or 3 times more abundant than the mouse that had the 131 reagent. And we actually report everything as a ratio to the pooled reference. So I told you, I don't know which one of these. Let's say this white one was the pooled reference. When the data comes out, we would say this peptide was measured in a mouse derivatized with the 130 reagent, and the ratio of 130 to 126 was like 2 to 1. And you could say another mouse derivatized with the 131 reagent, it had about a 1 to 1 ratio with the pooled reference. So everything is reported as a ratio against the pooled reference material. Okay, so take a break from that for a minute. And so I spent 3 years writing a grant to get this instrument. So it's the first of its kind in Texas. And it's called a Orbitrap Elite from a company called Thermo. And this is some propaganda about how wonderful this instrument is. But the bottom line is, when you do tandem mass tagging and those reagents I just described, you have to have a very good mass spectrometer to measure things to the 4th decimal place. And you have to collect lots of data fast. So we try to get MS-MS spectra as quickly as we can so that all the peptides coming off the capillary LC column are sequenced. If you had a very complicated mixture, it's hard to capture them all. So this instrument will show you some of the principles involved in liquid chromatography, tandem mass spectrometry. So this is, imagine the end of the LC column, and we ionize or make an aerosol of all the molecules, and then the peptides would come through the instrument, and we focus them to try to collect the things that we want to sequence and throw out the things that we don't. And this is what's called a Fourier transform tandem mass spectrometer. But it's a special kind. So it used to be that you use magnetic fields to store ions and make their measurements. And these instruments actually use electric fields. It's an Orbitrap instrument. So the ions are focused, and let's say we wanted to measure methionine and Keflin, we would say, just focus the ions with a mass of 574 Daltons. And it'll be stored in this, what's called the C-trap, and then we might break them into pieces to try to sequence a particular peptide. So this is the heart of the instrument. This is the Fourier transform part of the instrument. The peptide ions precess or move back and forth in an electric field. And as they precess, they do this at a certain frequency, which is dependent on their mass to charge. And because you can measure the frequency of things very precisely, we can get very good measurements of the mass to charge of the peptides, as well as the pieces of the peptides. And so this is, again, examples of the MS-MS spectra that you get. And we're collecting these really fast. It's like every 100 milliseconds or less we collect an MS-MS spectrum. So despite all this technology, it's still, I don't know, 10 or even 100 times slower than a lot of genetic techniques. But this is where we are with proteomics. So it's sensitive, it has a good dynamic range, fairly reproducible. This is a picture of my lab. The quality of the lab and the instruments look a lot like the lab here. You know, you have good instruments here. To do quantitative work, though, you do need some kind of Fourier transform or more expensive mass spectrometer to do like a large number of things. But maybe less complicated samples or more sample fractionation and preparation, you can still do a lot of nice work. So that peptide I talked about before is shown here. This is the sequence for serum albumin. And so you could say in this data set, we sequenced and identified that peptide. And if we saw the tandem mass tags in the reporter ions, we also quantified it. And so how do we quantify a protein? Well, we quantify a protein by measuring lots of different peptides within that protein. In this case, I don't know, 6 or 7 peptides were identified and quantified. And then you use an algorithm to try to construct how much, how does that reflect the level of the protein? So you could say if this peptide was, twice as abundant in the mouse that got labeled with the green reagent than in the mouse that got labeled with the white reagent, then probably all these others should also be twice abundant. But it's not that simple. So if they were all twice as abundant, you could say the whole protein was twice as abundant in the mouse that got labeled with the green reagent than the white reagent. And so this is what's called the protein inference problem. They had the same problem in genetics. You can think of it as a shotgun sequencing or shotgun proteomics. If you break the problem into lots of little pieces, it's easier to tackle any little piece, but it's hard to reconstruct the whole puzzle. And so a lot of times we will actually just report the peptide results and not try to infer the protein results. So if you imagine this is the EAE score that I showed earlier, right, where the disease peaks at about day 20. What you can see is sometimes we have biomarkers that go down with time. So this biomarker had a p-value, less than 1 in a million or something like that. And you can see that it actually mirrors the disease course, where it's inverted. So you see at day 20, the protein or this peptide went down in levels, and then it went back up at day 25. So this is a useful biomarker because it exactly mirrors the disease course. And you have others that actually go up with time. And so this one, it kind of follows along, and then it peaks at day 20 and then comes back down again. So we're actually not really interested in either one of these types of biomarkers this year. We're focused mostly on what are things that changed at day 7, things that showed a peak here well before day 10. It can either be a peak or a valley. Day 10 is when you start to make measurements of the EAE score. So we're interested in proteins that we can measure and assign to disease before the researcher can by just measuring their levels of paralysis. And you can correlate these either to disease score or to time. We actually get very complicated profiles. So even with just 8 time points like shown here, some proteins will peak 3 times just in 30 days. And that's something that's often overlooked in biomarker research, is how important it is to do longitudinal studies. So it's hard to get hundreds of samples to do some kind of study for a particular disease. It's even harder to get 10 samples from one patient. And so that's unfortunately what we need, in my opinion. So a lot of the work that we've done shows that it really matters, right? The time course really matters. So if you're a patient and you have multiple sclerosis, of course the physician doesn't know if you're at the equivalent of day 10 before clinical symptoms happen, or if you're actually, going to go into remission. You don't know what phase the patient's in. So you actually need to show the trajectory or the velocity of the protein. Is it rising? Is it falling? Is there a peak? And what does that indicate? So all the stuff we're proposing this year is actually longitudinal studies or trajectory studies. That was important, actually. I think 2 years ago they showed that PSA, which is often measured for prostate cancer, the levels of PSA are far less important than the velocity, right? Is it rising or falling in levels, and how quickly? So another thing that we do is what's called hierarchical clustering. So if you take all the protein expression data and you map it to a computer and say, just try to cluster the mice based on their protein expression. And I'm not going to tell the computer whether they had a lot of disease or a little disease, controls or what. So this is called non-supervised hierarchical clustering. What we found is all the mice with the worst disease clustered to the far left, and all the mice that had almost no disease clustered to the far right. So it basically worked even with a few samples. So I think we did 32 mice in this study, and the computer algorithm was able to take all the worst EAE scores and put them in the left and all the best in the right. And if you look at these branch points, there's about 4 branch points here. And so we just said this is the worst disease, the highest risk group, medium, and then another medium, and then finally this is the least risk group. So I study childhood leukemia too, and in a lot of diseases, like in childhood leukemia, what the physician tries to do is stratify the patients or put you in a risk group so they know how to treat you, because their protocols were developed to treat a particular risk group of patients. And so a common theme in the literature is to use omics technologies to get better stratification of patients and get ever finer, more personalized medicine. And so we found that with mice, you could do quantitative proteomics just like you can do these types of experiments with microarrays to stratify the patients, in this case, sick mice. So we've also done this in leukemia patients since then. And that finally takes us to step 6. So what do you do with the data? So quite often we'll get several thousand protein levels across 50 to 100 mice. And so it's a lot of data. You know, most grad students just, it's too much, right? So for the first year, you might be looking at the data. And that's another common problem in proteomics is you return the data to a researcher. It's like a long list of proteins from a few samples. Well, now we're giving a medium to short list of proteins, but over hundreds of samples. So what we found is incredibly important to make it easier to understand by mapping whatever we get onto a database of all known signaling pathways. So what can happen is then you can provide a cartoon like this to the researcher. This shows cytoplasmic proteins are shown here. These are cell membrane proteins, nuclear proteins. So you can get an idea of, you know, how did your sample prep work? Did you capture proteins from all over the cell? You can also map to particular protein networks. So this is the 14-3-3 signaling pathway. And this cartoon simply says there's 120 known proteins in this pathway, and we measured about 20% of them in this experiment. So you could say that pathway is enriched in this sample. And so that's something that we published. We found that there's a particular protein in the 14-3 signaling pathway which is involved in reactive oxygen species scavenging. And like in multiple sclerosis and a lot of brain injury diseases, that's very important. So this is a potential biomarker for the amount of ROS in your brain and your brain's attempt to clean up the reactive oxygen species. And so quite often in a project, we will provide something like this at the end of the manuscript or the end of the grant so that you can focus. So let's say that we've generated 2,500 proteins. Your next question is, well, how do I prioritize what to look at next? And so usually you'll pick something you're familiar with, in this case like myelin basic protein. But what's next after that, you might look, okay, well this pathway was enriched, so let's focus on 14-3-3. So it provides some direction to the biomarker research besides just the p-values or the fold changes. And the way that we prioritize our markers is we basically say the p-value is the most important thing, the statistical correlation to whatever we're measuring, in this case time or the EAE score. The second thing that we think is the most important to prioritize is the fold change, which you could think of as the ease of measurement. A lot of people flip those two. They'll try to go after the biomarker with the biggest fold change, even if it's a terrible p-value, because it's the easiest to measure. I think that's the wrong approach. So even if a protein just changes at 10% in levels, but you can measure that 10% change and it's a good marker, I think that's the one to go for, not the one that changed 20-fold with, you know, lots of error, 400% error. And then the third thing that we prioritize on is these pathways. So in my role as a core director, also when I'm just doing my own research, it's important to think about how long does it take to generate this data, take it from, you know, conception to publication, and then grant submission. And so we issue a lot of internal and external small grant awards. These typically take me about 16 hours to discuss with the investigator, figure out the best method, and then we will prep the samples. And because of our method, it says here 32 hours, but it's much, much shorter than that now with the M2 proteomics. Data collection, about the same as sample prep these days. Because we've multiplexed the samples, we already had a 30-fold improvement in sample preparation using the microwave. And then you can think, if you mix 5 samples together, that's times 5. So now you have 150-fold improvement in the throughput of the lab. This is actually our current bottleneck, statistical analysis. So the statistical methods for microarray are pretty well developed. So we're trying to borrow those. But the statistical methods for proteomics and this protein inference problem haven't been resolved yet. So we're doing our best to borrow from the microarray field. Some of the things that we do, for example, is this technique called quantile normalization. So you have a large amount of data. You can put all the data together and say, well, overall, all the mice that were labeled with the green reagent, for example, had a plus or minus 5-fold was their maximum protein fold change. And maybe all the mice labeled with the white reagent might have had a plus or minus 6-fold protein change. In the microarray world, they will normalize all the data, so it's a maximum of plus or minus 5.5. So we're doing stuff like that to try to normalize the data and account for some of the experimental variability. This is pretty quick, and then still the writing is slow, right? So the good news is we get a project, this particular project, we spent about 200 hours, or 1.25 months, to get everything ready for a manuscript. But we did a good job of it, and this person already collected data for 4 more manuscripts. And that's typically the way it is. So if you can provide some interesting data that has some proteins that the researcher is familiar with, it's pretty easy for them to write the story. But quite often what happens is it's so much data, it's overwhelming, and you don't know where to put your focus. And the technology that I described to get to this point, it requires a lot of expertise, and it takes a lot of patience to get good data. So I think it's worthwhile to spend a lot of time looking at the proteomics data and writing up a strong manuscript. But there's practical limitations. So a lot of the work that we do, for example, it's a small grant project. The person might have submitted only 20 samples. And so we're stuck with, for example, 4 mice per group, 5 groups. It's hard to get good statistics with 4 mice per group. We do our best, and we try to publish what we can, but it's definitely a smaller list of interesting proteins than we're capable of generating. So that usually generates a larger project, maybe 50 to 100 mice, and finally one of my researchers were up to 300-400 mice per project. So now we're starting to talk about the types of experiments that people do in microarrays, and also the types of things that are done in clinical studies. And so I was talking earlier with the proteomics core here, and I think that you guys are going to make an attempt at doing some microwave type sample preparation, which will speed things up. This is a presentation, actually, a slide from last year. But last year, I think we had 9 of these small grants, so maybe 9 small projects with 20 or so samples or less. Many presentations, probably 10 or so papers and grants a year. And this is reasonable for a core with 2 to 4 people. And it's still a tremendous amount of work. And so I think that it's nice to have a platform. You have to make some decisions. So unlike microarrays, there's really no standardization in proteomics. So each lab preps samples a different way. And if you change just one condition, if, for example, I use C8 magnetic beads versus C18 magnetic beads, which was a question from the students, yes, I'll get a different set of proteins. If I just, the moon is full, I'll get a different set of proteins, right? So the trick is to measure everything simultaneously so that you get good relative quantitation data. But you can make really subtle changes. This company, Waters, just changed the pH of their separation, and they get a completely different set of proteins between pH 2 and 4. And so a lot of times when we start a new project, the person's first question is, how many proteins can you measure? And I say, how many samples do you have? And they say, can you measure this particular protein? So we ask 3 or 4 questions with neither one of us giving any answers. Because I can't respond until I know, do you have to measure that protein? What is its level? How many other proteins are there? It's really a lot of give and take. To design a good experiment takes some patience, but I think it's worth it. And then another thing to think about is that, unlike a lot of the standardized methods for DNA or RNA work, everything matters. So the way the samples are stored in the freezer, proteins are very sensitive, right? And so you want to be very precise. Whatever you do, it's okay to do that thing, but do it across all 20 samples the same way every time, and then provide them all as a batch. Because we see a lot of artifacts which are really just, people are new to the field, and it's a little bit of a learning curve, right? So for example, almost all proteins have methionine residues, which are oxidized in the air. And so, if you can, it's best to cover the samples with argon before you put them in the freezer, especially if you're interested in methionines. Luckily, not too many people are, so you see in proteomics oxidized methionines everywhere. And it's nice because this technique can actually see those differences. So an oxidized methionine has a mass shift of 16 Daltons, so it's easy. When we establish the sequence, that'll always be 16 Daltons heavier than an unoxidized methionine. So that's the strength and the weakness of the technique. We see everything. So a Western is blind except to the protein you're measuring. We see everything. Sample prep, there's this one researcher who can tell which one of his grad students prepared the sample from skin contamination, from their keratin pattern. It's a fingerprint. So you need to be careful when you're doing sample prep. So that wraps up. This is from our RCMI conference. Was that in Nashville, I think? Nashville. And I hope I've convinced you that I know what I'm talking about. that Tandem mass spectrometry is sequence specific, and that's an important thing to remember when you're doing protein studies, not just to use Westerns. And there's a lot of analytical challenges of doing capillary LC-MS, which unless you're doing it yourself, you might take it for granted. And so this is very difficult to do well, but when it's done well, it provides a lot of powerful data. We have many putative markers of multiple sclerosis that I can't discuss, but we're hoping these make it through their way into a clinical trial someday. But it's a lot of work, and we're just now looking at patient samples. And then lastly, this technique, microwave and magnetic proteomics, I think you can probably adapt this for almost any method in your lab. Just take some magnetic beads and a microwave, and anything that you're doing that involves some chemistry and an aqueous buffer, you can probably speed it up. And so that's true for Westerns, for proteomics, for a lot of DNA and RNA techniques. And some things it doesn't matter, but for projects like this, it really helped us to take a year off and just say, we're going to reinvent this. So my lab no longer does gels at all. And that's okay, because any little condition changes the proteome that you measure. And we're really just interested in how does the proteome that you measure change across different mice or different samples, right? And that's it. I'll acknowledge all of these folks, and thanks to Loida for inviting me out, and for the core for showing me around. So thanks for your time.